Okay, we need to talk about gels, and it's gels of the electrophoresis. Thank goodness. We are one step closer to the actual lab and the importance of doing it. And the gels are important because the gels will hold the samples that I am preparing. So the samples that have already been replicated, that have already underwent the PCR, that's already did all of the cleanup, all of that is just given to me, and I'm going to take that for granted, and I'm going to put those into a gel, and I'm going to run them. So in this video, we need to talk about what these gels are and how to make them. And it's very simple, folks. There's nothing to it at all, I promise. If you can make Jell-O at home, you can make gels in the lab. So the gels are actually agarose gels. And agarose is just a powder that has to be dissolved. And it takes a little bit of heat in order to do this. But the nice thing about it is that agarose is very stable at super hot temperatures. So what goes on is that agarose is the common, not the only, but it is the common go-to to make gels for electrophoresis. And agarose gels, once they are poured and once they made, I want you to think of as like Swiss cheese. So we get these openings inside of the gel. Now you will not be able to see that, right? To you, it will look jelly-like. To you, it will look like Jell-O. It will be very flimsy. It will be very finicky. If you're not careful, it can break easily. It will be very moist and watery looking. I mean, that is a proper gel. But if you zoom in on the structure of how this gel is made, you're going to see really big openings and really tiny openings within this Jell-O-like material. So we're going to talk about those openings in just a minute, but that is kind of how this gel is structured. So the agarose is a powder, and that powder is going to get dissolved, but it's going to get dissolved most of the time in buffer. We never use water. It's got to be buffer, and the buffer has to match what goes into the actual electrophoresis chamber. That's very important, all right? We want everything to be consistent. We don't want changes. We don't want variables to take place. So the buffer that I'm using in the actual chamber itself, that is also the liquid that I want to use to make the gel out of. So I'll take this agarose powder and I'll mix it with some buffer. And then actually the only thing I have to do is just microwave it. I don't even need a hot plate. I can just microwave it. So put it in the microwave, close the door, put it in there for like 30 seconds. Take the flask out or the beaker, swirl it, see if everything dissolves. If it does not look like water, if it's not clear all the way through, if you still see like powdery looking stuff on the inside, put it back in. Put it back in for another 10 or 15 seconds. Take it out, swirl. Put it back in, take it out, 15, 20 seconds later, swirl. You keep doing that over and over in short time bursts until that agarose fully dissolves, all right? Once it fully dissolves, we are then going to pour it into something that we call a molding tray, and we allow it to cool. And when it cools, again, it will set up like Jell-O, and you'll get this little wiggly thing that begins to happen, and that is where eventually our samples are going to go into, all right? So those openings of the agarose is what allows electrophoresis really to take place, which is what affects the separation that takes place. So these really large openings and these really tiny openings, remember, samples are going to be injected up here at the top. And those samples are a mixture of stuff. And that could be really big blobby things. And that can be really teeny tiny things. Right? So that mixture is going to go into the electrophoresis gel. And that agarose is there. And the big blobby things begin to go through the big blobby holes. And because it's larger, it takes a little bit more effort to get it to move and shoving it through the holes. So it takes its time. It's a little bit slower. So the big blobby things are going to be slower and they're harder to move. And then the little teeny tiny things are going to go in and pew, 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 go through all of these holes easily because they can. Those are really big openings for them. They don't have to squeeze through them, and they can get in and out very quickly. So when the separation takes place, big blobby things are up at the top, and little teeny tiny things are toward the bottom. That's how this is going to work, and that's how this separates. So again, it separates based on molecular size, or in other words, molecular weight. So I say molecular weight, but really it is the molecular size. But these two things go hand in hand most of the time. Big, bulky molecules are typically much heavier in terms of molecular weight than smaller molecules are with smaller molecular weights. All right, so bigger takes longer to move. Bigger stays toward the top of the wells, is what we call those, where we inject our samples. All right. As far as DNA goes, DNA, which is kind of what's leading up to this point, that's what we're going to get. It has been RNA to DNA transferred over. That DNA, the way that it's structured, the way that it's made, it is a negatively charged species. And it's negatively charged because of two things. Number one, the sugars that make up the DNA. These are what we call sugar backbones. And those sugar backbones have OH groups all over the place. And those OH groups carry a very polar electron pair rich area, which means the entire backbone of DNA can be found as negative areas, especially when we begin to chop them and cleave them into smaller pieces or bigger pieces. The other thing that makes it negative, not only is it the OHs in the sugars, but it's also the phosphates that make it up as well. And those phosphates also have oxygens on them because they're phosphate. H3PO4, PO4, oxygens, more than one, also have free pairs of electrons. So those are also going to be very negative rich electron pair regions of the molecule that also makes it and helps it carry a negative charge. So we've got really big bulky things that carry negative charges, and we've got little teeny tiny things that carry negative charges. And what we would want to do is we would want to put a positive charge at the bottom of this gel. This is electrophoresis. So what happens is that this mixture of stuff that carries negative charges due to the sugars and the phosphates sees the positive charge way over yonder. And they want to go way over yonder. And in order to do that, they got to move. They got to get in the car and they got to go. They got to go. Got things to do. And the big bulky things take a little bit longer to get way over yonder. And the little teeny tiny things can get into the car and pew, off they go. So they travel at different rates and they begin to zoom down the electrophoresis gel. Again, this is a positive area. That positive area is often in electrophoresis chambers designated with the red cable. That red cable is positive charge. The negative pieces are attracted to it, which means that this is the anode. That anode attracts negatives. That is the term anode. And these are negatives that we have in the gel that we want to lure to the bottom. So we get movement. This DNA will pass through the openings of the agarose gel. The openings are different molecular sizes for different molecular pieces and fragments that are in our mixture. Those fragments will begin to go through the electrophoresis gel and migrate at different rates. The larger molecular weight fragments stay up on top. The smaller molecular weight fragments will zoom through the molecular sieve until it makes its way closer to the bottom, and we cut off the electricity of the electrophoresis before everything reaches the bottom. Otherwise, they will run off the gel and into our buffer, and we do not want that to happen. We visually see the movement if we're careful and clever about it, because what happens is that if we put what we call a tracking dye inside of our samples, we can visually watch these samples separate over time. And the tracking dye will show us, all right, so here's where we'll start. And we apply voltage or electricity onto the gel, the agarose, and we create this positive charge at the bottom, and then pew, straight through. So we could see maybe a band that shows up here, and a band that shows up here, and a band that shows up here, and maybe a band that shows down here. And then sooner or later, what happens is that the initial area that I injected that mixture into, that will go away. And that's because all of my pieces have now migrated. All of my pieces have left the point of origin. All of those pieces have moved through the gel and getting attracted to that positive charge at the bottom. And they're all different sizes, so they kind of hang out together because they all have that in common at least. But they migrate at different rates depending on the size. And then here I get like 4 chunks of material. I get a chunk at the top, and then another one below that, another one below that, and another one below that. And this up on top is going to be my larger molecular weight, and this will be my smaller molecular weight. Well, we don't really talk about molecular weights in terms of these. That's kind of difficult. But what we do talk about are something called base pairs. And the more base pairs we have, the bigger it is, and the more molecular weight it is. So they go hand in hand, but that's kind of our unit that we like to discuss when we talk about these fragments and how big they are. This is this many number of base pairs, and this is this many number of base pairs, and that's that many number of base pairs. What base pairs are, I don't even want you to know that yet. That, again, is for forensic chemistry, not here. It's just the stuff that makes up that fragment. That is what that is. That's all you need to know. All right. So we can watch this migrate and move in the gel while it's running. All right, so with that, let's kind of take a look at a better image of what I would normally see, and it would look something like this. So in lane number 1 that I see up here at the top for A, in lane number 1, a sample has been injected, and that sample's got 1, 2, 3, 4, 5, 6, 7. 7 bands in total. 7 chunks after the replication took place. 7 chunks of stuff. It's a mixture. So we're separating that mixture. And we're looking at how that cleaved. We're looking at how that replicated. We're looking at what's there. In lane number 2, very similar, we injected a sample. This sample is different because it doesn't match the banding that happened in lane 1. Totally different sample, right? So here I get 1 fragment, 2 fragment, 3 fragment, 4 fragments. 4 fragment pieces, 4 different sizes, 4 different number of base pairs. They migrated at different rates all the way down. And it's not the same sample, because if it was the same sample, it would perfectly match lane number 1. That's all electrophoresis is, ladies and gentlemen. That's it. Nothing more. Separation took place by charge, migrated through jello-like material, and we get separation based on the number of base pairs, which is kind of hand in hand to size or molecular weight. What you're seeing over here on A is just what I would call the raw gel. So this is the gel as you've made it. You're injecting samples. You're running it. You might see the bands, but they could be very faint or maybe a little difficult to see. That's all right. In part B, this is the same gel because if you look at the banding's the same in A and B. And the only difference is that this is a stained gel. And we have to stain it in order to visualize it a little bit better. So if these bands are really faint to the naked eye with just visible light, then we've got a few options of staining it and making it turn color. Blues or oranges or yellows or greens. Just really depends on what we're using. And very often those make the bands show up a little bit better. And that's what B is. So after you get done with electrophoresis, there is very often a staining step that takes place. And that allows you to visualize the bands much better. But if staining needs to happen, then you have to destain as well. And destaining is actually destaining the gel, not your sample bands, but the gel. The gel normally does not hold the dye or the stain very easy or very hard. It just releases it easily. The bands, though, of your DNA in this case, will latch on to the colored dyes, and it will normally keep them, and they'll be trapped. It will be very hard to get those dyes out of those samples or those fragments. So that normally stays. So what happens is that if we take something that's really faint, and let's say we stain it with a blue dye, that blue dye gets inside of those fragments of the base pairs, and it somewhat permanently attaches in there, and it makes it dark blue. And then it also gets inside the agarose, yeah, it does, and it will turn that blue. But the destaining process is normally just water, and water will help flush and rinse the dye out of the gel, but it can't really do anything for the fragments. It can't rinse the dye out of the fragments because they're in there and they're in there to stay. So this leads us into really now more formally the sample overview, like what's actually going to happen, or experiment overview. What's going to happen? How do all these like random pieces work together inside of the lab activity that you're going to be doing? So first off, the way that this lab is written is you've got 3 patients that you want to test. Do these patients have COVID, or are they negative? They might feel sick, but it might be due to COVID. It could be due to a different strand or a different virus that would carry different genetic material and not necessarily the N protein that the COVID-19 virus carries. And we need to figure that out. So they've already been swabbed. Those Q-tips have already been in the lab. They've already been extracted. They've already underwent the RNA to DNA transfer, so reverse transcription. And they've already underwent PCR, the polymerase chain reaction. All of that's been done. So someone else has done that work, and they've given me these samples, and they're saying, put them in electrophoresis and run them. Let me know what you find out. So what I'm looking for as a user, as an analyst, I'm looking for 3 bands. If one of these patients have 3 bands, then that means they are positive for COVID-19. And the reason there's 3 bands is because, remember, 2 of them are from the N protein, because we used 2 primers there, and I'm going to get 2 signals, each from that primer. The 3rd primer was an internal control. It was something that was supposed to show up every single time, no matter what. So a positive COVID-19 person will have 3 bands that appear, 2 from the N protein and 1 from the internal control. If the person's negative, then what we will see is one band, and that's because that band has came from the internal control. That is a self-check mechanism that we've put in place to make sure that everything was processed the right way, sample was injected the right way, PCR happened the right way, we injected it into the electrophoresis the right way, we developed the gel in the electrophoresis the right way. That is what this is all about. So we should see one signal if that person is negative. But here's the key. If this internal control is not there, and I don't see it at all, no matter what else is there, no matter what else is present, if that internal control is not present, then that means the test is going to have to be deemed as inconclusive. And it's got to be reran. It's got to be done again. Because that internal control failed. And that internal control, if it fails, means that there was a problem somewhere. Either in the swabbing, or the replication, or in the transcription, or in the sample injection, in the electrophoresis, in the staining. Something has went wrong for that internal control not to show up. And if that is not there, even if other bands are present, if the internal control is not there, we cannot make a judgment call. We cannot say negative. We cannot say positive. We have to say inconclusive. And that means the test is repeated from the very get-go, from the very first step of swabbing. Or maybe not. Maybe if they got enough, maybe they can start from the PCR side and try it again. But if they're unlucky, yeah, it's got to be swabbed again. It's got to be reran. Things have to be added to the original sample in order to replicate. The way that these are going to be set up, when you get your strip, these are all going to be sealed into an aluminum strip. Those strips are going to be labeled as A, B, C, D, E, F. You'll see that stamped on the top. Tube A is going to be what we call a standard marker. I'll talk about that in just a minute. Lane B is a negative control. That means this is what you would see if that person does not have COVID. That should be one band, the internal control. That's it. Tube C is the positive control. That means if the person is positive, this is what you will see. And that will probably have 3 bands. 2 from the N protein and 1 from the internal control. And then tube D, E, and F is going to be regarded as patient 1, 2, and 3. Are those patients positive? Are those patients negative? Are those patients inconclusive? Because they all should have 1 band at least. And that 1 band should be the internal control. And that 1 band should match tube B, which is the negative control. They all should at least show that one. Every single one of them. If they have that missing, it is inconclusive. And you can't really tell them positive or negative. Also keep in mind that before you load your samples, make sure the gel is properly oriented. And what this means is that here's my gel. Here are the wells that I'm going to inject the sample into up on top or on one side. And these wells need to be facing the black power cord. And the black power cord is the negative side of the electrophoresis chamber. Keep in mind, these are negative-based fragments. And because they're negative-based, they are going to be attracted to the positive side. And they have to run this way. If you flip your gel in there backward, they go the other way and they'll run off the gel and into the buffer. You won't be able to do anything with it at all. So before you inject any samples, if you're unsure, let us see how your gel's oriented. We'll let you know if it's in there the right way or not. And if not, we'll flip it around. But your wells have to face the black cord. That's your negative side. And those samples will run toward the positive side, which is the red cable of the electrophoresis chambers. Please do not get it backward, otherwise it's ruined. All right, so next up, we're going to talk more about making this agarose gel. Well, kind of we already done that in a way, but I'm going to go into a little bit more detail with that. We know we got to weigh out some powder. We know it's got to get dissolved. We know it has to be microwaved. But I'm going to visually show you some steps about what the procedure is going to want you to do and how to follow through with that, making these recipes the proper way.